Fellow board member Roger Mail, distinguished guests, colleagues, ladies and gentlemen, welcome to Lecture 5 in the We High Public Lecture Series, the first for 2010. But let me firstly say I'd like to acknowledge the Wurundjeri people, who are the traditional custodians of the land on which we meet. I'd also like to pay respects to the elders, both past and present, of the Kulin Nations, and extend that respect to other Indigenous Australians present this evening. Now, my name is Graham Mitchell. I am privileged to be your MC, chairperson, moderator, whatever, this evening. Normally it would be, of course, the director of We High, Professor Doug Hilton, who would be standing in this spot. And so I apologise on his behalf. He's been called away and unable to join us this evening. And so my only credentials for being your chairperson this evening is that with one of the speakers tonight, I actually established the malaria program at We High, and one of the very first PhD students was another of our speakers, Alan Cowman. And James, I'm sorry, in this little historical anecdote, I don't know how to weave you in. You were probably in primary school or something at that stage. But Professor Hilton has asked me to spend just a couple of minutes. I know you have things in the bag, sort of telling you about We High, but just to say a few words about the We High research agenda. The Walter and Eliza Hall Institute of Medical Research is home to more than 550 researchers who are working to understand, prevent and treat diseases including cancer, particularly leukemia, lymphomas and breast cancer, type 1 diabetes, arthritis, celiac disease, HIV, hepatitis and malaria. Now, We High is keen to see its discoveries and inventions translated into new medicines. And in that vein, at the current time, there are 30 clinical trials based on discoveries made at this institute that are currently underway. These include trials of vaccines for type 1 diabetes, celiac disease and malaria, and trials of a new class of anti-cancer drugs for treating patients with leukemia. Now, with those 550 researchers and the research budget of $75 million, you would expect some pretty major discoveries. Well, the news on this front is very good. In 2009, there were 3 highlight discoveries. The first in breast cancer, the identification, the discovery of a population of breast cells likely to be responsible for breast cancer that develop in women carrying mutations in the gene BRCA1. Secondly, in cancer biology, new information on what proteins are involved in cell death and clear identification of what proteins are or are not attractive targets to accelerate cell death in, for example, cancer. And in malaria, thirdly, the Institute researchers have created the world's first genetically modified strain of the malaria parasite to be used as a live vaccine against the disease. I'm sure we'll hear something of that this evening. And in the first 4 months of 2010, again, there are riches in this area. Firstly, the Tasmanian devil facial tumour disease, which you will have seen as horrific pictures of that disease, that sort of disfiguring, lethal, transmissible cancer affecting Tasmanian devils. And Australian researchers and We High researchers, in collaboration, have identified the cell type that is the origin of that transmissible facial tumour disease of Tassie devils. And that's the Schwann cell, which protects the neurons in the peripheral nervous system. And in malaria, We High researchers have identified a key protein used by the malaria parasite to transform human red blood cells and to promote its own existence in the body. And I'm sure we'll hear more about that today. Thirdly, the structure of the insulin receptor. We knew the structure of the receptor some years ago, But WEHI scientists have identified what portion of that receptor the insulin actually binds to. Crucial portion of the jigsaw puzzle, knowing what it is about that molecule, that where insulin binds, and therefore focusing attention on that area of the receptor, the insulin receptor, for sort of new drugs. And in breast cancer, to go back to the first discovery that I mentioned in 2009, WEHI scientists have discovered that these breast stem cells, the origin of many of these cancers, are exquisitely sensitive to female hormones, estrogen and progesterone, again throwing new light or further light on female hormones as risk factors in breast cancer. So that is a little snapshot of a very small portion, like going to a restaurant. You don't get the total menu in one setting, so you get little snapshots. And so that was a snapshot of some of the things which are happening in WEHI, which the director was very keen that I transmit to you this evening. But you're not here to listen to me. You're here to listen to authoritative voices in the field of malaria, a global disease, and control and eradication. So we have three speakers. The format will be that the three speakers will present. We'll leave discussion to the end. We will have a panel discussion, which I'll sort of moderate, where questions from the audience, questions amongst the panel, of course, sort of, and we'll have some in that grand Western tradition of putting the experts sort of under the microscope, and sort of and some really robust discussion, which there's a great tradition of WEHI, which I know Gus Noss will sort of certainly sort of fertilise. The sound up a bit. Shall I shout a little bit as well as the sound going up? Is that testing? Okay. So yes, let me know if there's an issue with respect to the audio visuals, for that matter. Okay.
If you want the mic, I can give it.
Well, the first speaker this evening is Professor Alan Cowman. Alan is the head of the Division of Infection and Immunity at the Institute here. He is the holder of a prestigious Australia Fellowship, a Senior Principal Research Fellow at the Walter and Eliza Hall Institute, and for most of his career, he has been interested in understanding the function of proteins in Plasmodium falciparum, a name we'll hear a lot tonight, the causative agent of the most severe form, the lethal form of malaria. He's made major contribution and is well known globally in the area of drug resistance, understanding the mechanism whereby Plasmodium resists that sort of armamentarium of drugs that we have, and the mechanisms, therefore, and looking for ways of overcoming that drug resistance. He's been a key player in a field which many of you may not have heard of, transfection technology, and we'll hear that tonight in his speech in a moment, in Plasmodium falciparum, a really major breakthrough technology in a deeper understanding of how proteins interact, sort of what's happening in the malaria-infected cell, and the role of genes in sort of promoting drug resistance, for example, and how the malaria parasite goes about its dastardly business in the body. His work has led to over 200 research publications and resulted in numerous local and national awards. Just before Alan comes to the podium, I should indicate that tonight's proceedings are being recorded and filmed, and they will be available. So those people who are going to ask questions realise it will be in perpetuity rather than in our memory box for just this evening. So without further ado, Professor Alan Cowman.
Thanks very much, Graham, for that introduction. It's fantastic to see so many people here. It really is. Welcome to WEHI. So what my job is to tell you a little bit about malaria, and then James and Gus Nossel will take it further. What I'd like to tell you about is firstly the global impact of malaria. I'll tell you a little bit about the history of malaria. and also a little bit about the malaria parasite and how it infects humans and how it causes disease. And then what I'd like to do is give you an example of some of the research that has taken place at this institute in collaboration with some of our colleagues in Melbourne, and show you how that tells us something about the parasite. So firstly, a global burden of malaria. And malaria is truly a global disease. This is a map of where malaria occurs, and you see by the countries that are coloured in red that it occurs mostly in tropical and subtropical regions of the world. Now, one country that is not coloured in red is the country that we live in, Australia. And a lot of the questions that I get is, why do we work on malaria when there's essentially no malaria here? Although there is malaria, people get malaria, usually travellers coming from overseas. And the answer to that question is shown in this list here. And that is that there are 3.3 billion of people exposed worldwide to malaria, over 250 million cases, and over 1 million people die of malaria each year. And most of that burden falls on pregnant women and children, particularly children under 5 years of age. It's responsible for 1/3 of preventable low birth weight babies, and that has a huge effect looking forward as these children grow up. It's a leading cause of childhood death. And if you look at Africa, they have 1 in 5 episodes of malaria per year, depending on which particular country within Africa they live in. Now, at the moment, there is no effective vaccine, although a lot of people are working towards that, including scientists here. And treatment with anti-malarial medicines is becoming less effective for reasons that Graham alluded to, which is the ability of the parasite to circumvent those particular anti-malarial drugs. You know, it's not just a disease problem. It causes huge economic burdens on the countries where it occurs. If you look at the numbers, more human hours are lost to malaria than any other disease. If you think about HIV and tuberculosis, more people die of tuberculosis and HIV than malaria, but more human hours are lost with respect to malaria, and that's because its biggest impact is on children under 5 years of age. When they die, they lose essentially the rest of their lives, and if you add up those hours, it is essentially more human hours lost than tuberculosis and HIV. The gross domestic product of countries where malaria occurs is decreased from 1% to 4% per year, and it's essentially a measure of economic growth. So in Australia, the economy grows in good years from 1% to 3%. So if you think about it, if you had, before you even started the year, you had 1% to 3% basically growth disappear because of a disease such as malaria, it really stalls your ability to develop for the country, and also stalls the ability of the country to provide the services that we expect here in this country. So it traps families and communities in a spiral of poverty, and it disproportionately affects marginalised and poor people. And there's a lifelong effect through increased poverty and also impaired learning, which is what I mentioned with respect to low birth weight, but other reasons as well. It cuts attendances at school and workplaces. And probably the most important aspect of this list is it is preventable and it is curable. So it's a disease that can be treated. It's a disease, if the political will and the scientific knowledge is there, it's a disease that can be controlled and perhaps even eradicated. And Gus Nossel will talk about that at the end. Now, when people ask me about malaria, when I say I work on malaria, usually they say, well, wasn't that cured a long time ago? And the answer to that is no. It certainly hasn't, and I've just shown you the numbers. It truly is an important disease and affects a lot of people throughout the world. It's an old disease. It's been with us for a long time. In fact, it's been with us through human evolution. And you can find words written about it throughout history, and perhaps one of the first is Hippocrates in 400 BC, who described the symptoms of malaria and basically observed that attacks of this particular disease seemed to be related to time of the year, wet season, and also appeared to be related to closeness to swamps. If you think about that, that's the time and the place where there'll be more mosquitoes, and we'll come back to that in a minute. And some of you might have heard some news clippings that described some recent findings where they analysed the body of King Tutankhamun and were able to detect malaria parasites in Tutankhamun's bones, essentially. What that suggests is that Tutankhamun had malaria. They suggest that Tutankhamun died of malaria. It's possible. Since it occurred 1,324 years or so ago, it's hard to say that he actually died of malaria, but he certainly had malaria when he died. Now, if we come a little bit towards the future, to the late 1800s and the discovery of the malaria parasite. So before that, everyone knew about the disease because a lot of people were getting malaria. They knew that it seemed to occur in rainy seasons and that if you live near swamps, you seem to get more malaria. So in the late 1800s, there was a lot of people trying to find the causative agent of malaria. And the person who won that race was Alphonse Laveran. Now, this is in the time of Louis Pasteur, who basically espoused the germ theory. So there were a lot of people who believed that malaria was caused by bacteria, and they were looking for bacteria as the causative agent of malaria. In fact, it's not a bacteria, and I'll go into that in a little bit. Now, these are drawings that Laveran actually drew back in the 1880s. And were published in one of his papers describing the malaria parasite. And he discovered the malaria parasite in Algeria by looking at blood from patients that had fevers that was symptomatic of malaria. And what he identified was a parasite, a protozoan parasite. A protozoan parasite is very different from a bacteria. Bacteria doesn't have a nucleus. Bacteria has a cell wall, whereas protozoa has a well-defined nucleus. Nucleus contains all of the genes and DNA that essentially will make a new parasite. And it doesn't have a cell wall. So it's a much more complex organism compared to a bacteria, but it is single-celled as a bacteria is. Now, remember, when Laveran did this, he won the Nobel Prize in 1907. And when he did this work, we didn't have the ability to stain the parasite, to visualize it in the ways that we have today. So he was looking down a microscope, trying to see the malaria parasite in the blood of patients. And this is what he saw. So this is a red blood cell here. This is a parasite inside the red blood cell, a malaria parasite that is moved into the red blood cell, and I'll show you how that happens later. And some of these are the different forms of the parasite as it grows inside the red blood cell and divides and makes new forms of the parasite. Now, this is a stain and smear from essentially a manuscript published 5 years ago. So using the techniques we can use today, where you can stain the parasite with a stain called Giemsa, see it inside the red blood cell, and very easily see that that's something that shouldn't be within a red blood cell. Now, if you look at that, you see a nice round circle that looks like a ring with a nice punctate dot here. Looks like the diamond of a ring. In fact, it's called a ring stage for that reason. Look at Laveran's drawing here. You see a beautiful ring with a nice punctate dot here at the top. And here's another one just here. So he got it pretty well right. A very important discovery. Now, once the malaria parasite had been discovered, there was, again, another race to try and understand how malaria is transferred from human to human. And some people believed it was a mosquito. And in fact, the person who showed that it was a mosquito was Ronald Ross. And he also received the Nobel Prize in 1902, actually before Laveran did. Both discoveries are just important. Here's some drawings of Ronald Ross. Clearly not as nice a drawer as Laveran, but this is really his lab book, just noting down some of his work as he goes along. Now, he started this work in 1895 to try and prove the hypothesis that was set up by Laveran, but also Patrick Manson, who was really the father of parasitology, an Englishman. And basically try and prove that mosquitoes were connected with the spread of malaria. In 1897 in Secunderabad in India, Ross made his landmark discovery by dissecting the stomach tissue of a particular type of mosquito, Anopheles mosquito. It happened to be a female Anopheles mosquito that was fed 4 days previously on a patient that had symptoms of malaria, and he found the malaria parasite, and he went on to prove that it was this particular type of mosquito that spread malaria. Now, that kind of sounds like there was one day where he had a eureka moment, discovered malaria parasites in insects. It doesn't work like that in science. Basically, what this represents is a number of years of hard work basically observing and dissecting mosquitoes and looking at transmission of malaria from those mosquitoes. Okay, so this is what Anopheles mosquito looks like that is obviously fed on a human. The blood that you see inside the mosquito, the blood that squashes out when you slap it on your arm is not the mosquito's blood, it's your blood. And this particular mosquito has fed very well and its gut is full of blood. Now, as I said, malaria is a protozoan infection. It's transmitted to the human beings by the female Anopheles mosquito. Now, just in case you think there's only one type of malaria parasite, that's not the case. There are at least 120 species of malaria parasites called Plasmodium. Plasmodium falciparum, as Graham mentioned, causes the most severe form in humans and causes almost all the deaths in malaria. Now, it's interesting that all of these species of Plasmodium infect organisms from lizards to birds to monkeys to humans. So just about every organism has its own Plasmodium parasite. Probably dinosaurs were infected with Plasmodium as well, although there's none around to prove it. But I think it's probably likely. So there are other Plasmodium species besides falciparum that infect humans, including Plasmodium vivax. And when people think of malaria, they think of it coming back 1 or 2 years later. And that particular form of malaria is in particular caused by Plasmodium vivax. And there are 3 other species that can also infect humans, but it is Plasmodium falciparum that causes the vast proportion of the deaths. Okay. So what I'm going to show you is a couple of movies that Drew Berry, this is Drew, did. He does animation here at the Institute. He's won a lot of awards for some of this animation, and I think you'll see why when we look at this. What this shows you is the life cycle of the malaria parasite. And what I'm going to start with is the life cycle within the human host. So it starts with a mosquito biting a human. We're going to have sound effects here. So just imagine you're in Africa, near rainy season, lots of mosquitoes around, buzzing around, and there's one that you hear but you can't see, lands on your shoulder and bites you. This is a slow mosquito. Okay, so here it comes. It's going to land on the shoulder and bite. So it has a proboscis that it inserts into the host, and that's when it usually hurts, and that's when you usually slap it. Now, most people think that when it goes in, it actually finds a capillary and then sucks up blood. It's not like that at all. What it does is push its proboscis in and move it around and basically slash a lot of capillaries and cause a lot of hemorrhaging, so that it has a pool of blood that it can suck up. So we'll move into the human as the mosquito is feeding. So these are capillaries under the skin of the human that's just been bitten. It's infected with malaria. It has malaria parasites within its salivary glands. It injects part of its salivary gland, part of its salivary solution into the human to basically stop clotting of the blood. When that happens, some of the parasites move in and they're able to find and seek out capillaries so they can move through the blood system. They then move to the liver and they can sense cells within the liver because the liver cells that they want to move into, invade and grow. And you're going to see that happen in just a moment. Okay, so the parasites have moved into the liver, found the liver, and they start moving through liver cells. They're able to basically move straight through them. And the mechanisms of this are questions that we and many other scientists are trying to work out. Eventually, the malaria parasite finds a liver cell that it likes. Some of the cells that it moves through, it kills, such as this one, which is called a Kupffer cell in this case. It moves through this liver cell, and this liver cell dies, and then the next one it moves into, it decides to start growing and dividing. So from one malaria parasite, you get thousands of forms that are called merozoites. This slowly grows within the liver cell until liver cell is basically consumed parasite can move out of that cell. Eventually it bursts open, move out and into the blood system. Okay, so we see parasites moving into the blood system. So far there is no disease, no symptoms with respect to this infection. However, in the blood stage, it now invades and starts to grow inside red blood cells and multiply. And that's when you start to get symptoms of malaria, and you'll hear more about the disease when James Beeson talks. Now, it invades these red blood cells, which are the cells in the blood that carry the oxygen and carbon dioxide around the blood from the lungs and the tissues and back again. It sticks and identifies a red blood cell and then starts to move inside, seals the membrane around it as it moves in. Some proteins are shaved off that particular form as it moves in, and then it starts to grow, and this cell undergoes an amazing remodeling process where it converts this red blood cell into a cell that the parasite can grow, survive, find its nutrients, and also evade all of the mechanisms that the host immune system throws at it. So it's an amazing organism. Now, what it confers on the red blood cell is the ability to stick, to basically adhere to blood vessels. I'll come back to that because that's an important mechanism that the parasite uses to essentially evade mechanisms that the human host uses to try and destroy it. Now it continues to grow, invade, move out of the red cells, invade, and so there's a continuous cycle as it multiplies in the blood. So as I said, all of the symptoms are shown here from this stage.
Is the sound effects too loud, or can you hear Alan? Okay, all right.
So what you've seen is half the life cycle in the human host. There's another half in the real definitive host, the mosquito, and that's what we're going to see here. Again, a mosquito. This one's a little bit faster, buzzing around, and everyone's heard that noise. It usually happens if you're trying to go to sleep at night time, and that's what you're trying to do, is to squash it. So this mosquito has had a feed. This is a female mosquito, and it's only pregnant female mosquitoes that bite because they require the blood for the development of their eggs. So this one has had a blood meal. If we move into that mosquito, the red is all of the red blood cells, essentially of the human that it's bitten, that happens to be infected with malaria parasites. So the mosquito has taken up malaria parasites from the human host. Now we'll actually go back to the human host because some important events actually occur there, and that is sex. Malaria parasites do have sex, and we'll actually see that. It's not X-rated, but we'll see it. So this is inside the gut of the mosquito. Most of the malaria parasites die. It's only a few specialized forms that are developed as sexual forms, and these are, in the case of female ones, are large oblong cells within the red blood cell. So when the mosquito takes those up, because the gut is at a much lower temperature, this process of sex is activated. The female forms develop into eggs, whereas the male forms develop into essentially the equivalent of sperm. And what you'll see in a minute is a sperm swimming up here to fertilize this particular malaria egg. Okay, that's a slow one. It will burrow in, fuse with the egg. Their genetic material will fuse and be reassorted, and you will get essentially a fertilized zygote. This develops into a long form that becomes motile, that can also sense where it is and also find the place where it wants to go, which is outside the stomach of the mosquito. It burrows outside of the stomach and sits on the outside of the stomach, where it develops and divides into thousands of different malaria parasites. You'll see it come through so this is the lining of the stomach of the mosquito. It's coming out to the outside. It'll sit there and develop to form these forms here, which are called sporozoites. And these can also sense where they are within the mosquito, and they can migrate to the salivary gland of the mosquito, and these are the forms that are injected into the human host when the mosquito is feeding. So essentially that's the full cycle of the malaria parasite. It's quite complicated, but it's very efficient and actually really quite beautiful, particularly when you're looking at Drew's movies. Thanks, Drew. Okay, so with malaria, as I said, it's been with humans throughout their evolution, and you've really had this evolutionary arms race of the malaria parasite with the human host. Essentially, you've had each of them trying to gain an advantage so that one can infect the human host sufficiently, cause disease, but not too much disease, so it can actually get in there and be in there long enough to be taken up by a mosquito. And the human host that essentially wants to live and survive and basically have children and live a reasonable life. So there's a huge selective pressure, typical or very classical Darwinian evolution. And a really good example of that is sickle cell anemia or sickle cell disease. And essentially this is caused by a mutation in the hemoglobin gene of humans. Hemoglobin is the protein within red blood cells. It's the protein responsible for carrying oxygen, carbon dioxide from lungs to tissue and back. A very important protein within the red blood cell. It happens to be the major protein that the parasite utilizes in the red blood cell, because the red blood cell is essentially a bag of hemoglobin protein and not that much else. Sickle cell results from a mutation, a single mutation in this particular hemoglobin gene. That changes the shape of the red blood cell, so you see this sickle-type shape. This is the normal shape of red blood cell here. And when you look in areas where malaria is prevalent, you see that 33% of inhabitants of sub-Saharan Africa, for example, carry the sickle cell gene. So most people have sickle cell gene but don't necessarily get symptoms, but there are some that get severe symptoms, can die, and can be very sick. So there's a negative component of having this particular mutation for some people. But carriers have a reasonable protection against severe malaria disease. That means that they can live through childhood, that they can grow into adults, have children, and pass their gene on to the next generation. And so with that selective pressure, so that particular people with this particular mutation are more likely to survive in malaria disease, what you end up with is that particular mutation occurring within the population at very high levels. And this is the spread of people that have this sickle cell mutation. And if you overlay on that the areas where malaria occurs, you see almost a perfect match. So clearly malaria has been selecting this mutation, that it has arisen in humans to provide a selective advantage for survival against severe disease, severe malaria. Now, as I said, you've got the huge pressure on evolutionary pressure on the human to come up with mutations that give it an advantage, but parasite, you've got exactly the same thing. It's trying to come up with tricks that enable it to get around anything that evolution throws up against it in the human. So I just want to go through one of the tricks that the malaria parasite has evolved to survive in the human host. And in fact, there are many. And some of the research that we and other scientists are doing around the world is to understand some of these tricks, because they're absolutely key to developing new vaccines and new drugs against this disease. The malaria parasite invades red blood cells and it exports a particular protein to the surface, and Graham Mitchell mentioned this at the very beginning. It's one that's called PFMP1. This is on page 121 of the biology textbook for grade 12, for those who are doing grade 12. So it is mentioned in the biology curriculum for grade 12s. So hopefully you'll learn a little bit more about it tonight. It's exported out of the parasite. This is the parasite through the red blood cell, and then presented on structures called knob structures. And I'll show you what they actually look like in a minute. And what this protein is essentially a molecular Velcro. It's the protein that is on the outside of the infected red blood cell, allows the parasite to stick. And you saw the animation of infected red blood cells moving through the circulation and then sticking to blood vessels. This is the protein that does it. Why is this an advantage? Why has the parasite bothered to develop a system like this? The answer is it's no longer circulating in the blood, and it cannot be removed by the spleen. The spleen is an organ that occurs around about here in your body. It's a sentinel organ. It's looking for altered red blood cells, and you can't get anything more altered than a red blood cell with a big fat malaria parasite inside it. So if that moves through the spleen, it'll be quickly recognised, removed, destroyed. So it's a huge advantage for the parasite to stick in organs and so that it doesn't move through the spleen. And you'll hear a bit about that from James Beeson in terms of why that causes disease. So how do we know that? How do we know that? And I'll just run through very quickly through one experiment that was done here, in fact in collaboration with Brian Cook, who's actually here, and I'll show one of his nice movies of flow. And the reason we know that is because we and many other scientists have done experiments to prove it. And I'll just run through one experiment. This is a red blood cell infected with a malaria parasite. The malaria parasite has remodeled and altered this red blood cell and formed these knob structures. The membrane's usually very smooth. So these are the knob structures. On these knob structures is the PFMP1 protein, the Velcro protein responsible for sticking. What we've done is genetically modify this parasite. We've used molecular scissors, essentially, to go in and disrupt a particular gene encoding a particular protein that we thought would be involved in forming these knob structures. That means that you would have a infected red blood cell that's smooth, that has disordered PFMP1 on it. So if PFMP1 was responsible for stickiness, for the Velcro stickiness, this particular red blood cell would stick very well. This particular one that we've genetically altered would not stick, or would stick much less. And this, again, is a movie that Drew made probably 15 years ago now. Essentially, this just shows the transfection method that Graham alluded to at the very beginning, where we're specifically inserting a piece of DNA to disrupt the function of a gene and the expression of a protein. But the parasite is still able to invade red blood cells, can still grow in the laboratory, except it doesn't have knobs, and the PFMP1 on the surface is disordered. And what we would hypothesize is that it would no longer stick, or would stick much less well. How do you do that experiment? You can't. This parasite infects humans. You obviously can't do experiments on humans, so how do you actually prove that? What you do is you build a blood vessel in the laboratory, and this is what work that we did with Brian Cook, where essentially what we've done is made the equivalent of a blood vessel with proteins on that side, and flow infected red blood cells across it, and ask, do they stick? And this is a normal parasite, infected red blood cells, sticks very well, whereas this is our genetically modified organism, which doesn't stick very well at all. So what that tells us is that PFMP1 and those knob structures are really important for sticking, for that Velcro effect. And these are, in fact, infected red blood cells. This is what Laveran would have seen when he looked down a microscope. No stains. This is exactly what it looks like, a red blood cell, a parasite just in here. This one has two, with little dots spinning around. He wouldn't have seen the pixels, though. So, for those of you who can see this area and have been watching, might have noticed this little counter down here, and it's not counting number of slides. The World Health Organization official numbers are that one child dies every 30 seconds from malaria. So for the time that I've been talking, the equivalent of 32 children have died. Now that doesn't sound like very many, but when you equate that to 1 year, it's over 1 million. So over 1 million children die of malaria each year. So malaria is truly a global health issue, and it's one that we should be concerned about, even though there is no malaria in Australia. So thank you very much.
Thank you very much, Alan. A wonderful overview of malaria, the parasite, the infection, the biology of the parasite. We now move to a presentation from Dr. James Beeson on more the disease of malaria and why people die of this awful infection, with the numbers, the arithmetic which Alan has highlighted here. James Beeson trained in clinical medicine at Monash University, and undertook further training as a public health physician. He completed the PhD here at WEHI in 2000 on malaria in pregnancy, and moved to Kenya in 2002 to pursue studies on immunity to malaria. He returned to WEHI in 2004 as the inaugural Jacques Miller Fellow and established a malaria research group. He's a laboratory head within Alan's Division of Infection and Immunity. His research is aimed at understanding immunity to malaria, developing vaccines against this disease, and he's working with colleagues in Kenya, Papua New Guinea, Thailand, and Malawi, where malaria is a major health problem. James Beeson.
I'll try and use the podium mic. Let me know if you can't hear me. Thanks for that introduction, Graham, and to Alan, and of course thanks to all of you for coming along tonight. Let me just start this up. Okay. Yeah, so what I want to talk about now is really some of the clinical aspects of the disease, malaria, how people develop immunity, why people become ill, what are the sort of features that you see, and what's some of the work that we're doing here at the Hall Institute to try and develop new ways of treating and preventing malaria. So let's start with the clinical features of malaria. Most malaria in humans is actually a relatively mild disease, so it causes typically what we'd call a flu-like illness. So pretty similar to what you'd experience, once every couple of years, where you feel pretty sick with a virus. Fever, headaches, malaise, aches and pains. That's the majority of the cases around the world. So it's relatively non-specific. You can really only diagnose it by doing a blood test. In a proportion of cases, excuse me, people develop severe malaria, so life-threatening illness essentially. And a proportion of those who develop severe malaria go on to die, unfortunately, as Alan highlighted. These are the sort of main features that we see in people who develop severe malaria. You don't necessarily see all of these features in any one person. It's sort of an overlapping mixture of different features. But these are the most common. Severe anemia, so a severe lack of red blood cells or hemoglobin to carry oxygen around the body. Cerebral complications, so that's typically referred to as cerebral malaria, so affecting the brain. You get coma, convulsions or fitting. And of people who actually survive that, long-term neurological deficits are common that affect learning and development and function throughout life. Respiratory distress and metabolic acidosis are really important features of severe malaria. That's a little bit complicated, but respiratory distress is, you see in these children particularly, and true also in adults, very laboured breathing. It seems like the person's really struggling to get enough oxygen into their blood, and their heart's not pumping blood around the body well enough to get into the tissues. And metabolic acidosis is due to the whole picture, the impact of the parasites in the bloodstream that disturb the whole metabolic function of a person and their circulation, so that they get an accumulation of acid in the blood, and so their blood becomes acidic. There's also lung damage, which is affecting the ability to oxygenate the blood. There are a whole raft of other features that you might see in someone who has severe malaria, which include low blood sugar, hypoglycemia, kidney failure, or blood clotting problems. So it's really a complex disease, and it's difficult to treat, which I'll cover. So I thought I'd touch on why do people get sick with malaria? What is the process that's going on? I think fundamentally it results from this unrestricted replication of malaria in the bloodstream. So, for most of us in the audience, if we don't have any sort of immunity to malaria, if we got infected and we didn't seek treatment early enough, the parasites would get out of control in the bloodstream, disseminate throughout the body, and have a wide range of impacts that would lead to severe illness. I think a lot of us would end up in hospital, in ICU. So one of the effects is that malaria parasites start to accumulate in vital organs. As Alan highlighted at the end of his talk, the parasites that are inside red blood cells can adhere to blood vessels, inside blood vessels, and that enables them to accumulate in vital organs. So these are actually pieces of tissue taken out of the brain or the placenta and then viewed under the microscope. In this case, out of the brain of a child that died of severe malaria, and it shows a small vessel in the brain here. They're the vessel walls. And this should all be full of nice looking clear red blood cells, but it's not. It's full of these purple dots, which are all malaria parasites. And I think you can get the impression from that that blood vessel is probably completely occluded, clogged up with malaria infected red blood cells. And you can imagine the effects of having, hundreds, thousands of small vessels blocked by malaria parasites in the brain. It's disastrous. In malaria in pregnancy, I think Alan highlighted that pregnant women are at high risk of malaria, partly because of the appearance of the placenta required for fetal development. And parasites can accumulate in the blood spaces of the placenta. So this is the maternal blood, which sort of pools inside the placenta to allow the exchange of oxygen and nutrients to the baby. And so you can imagine all of these purple spots are malaria parasites. So they're just packed full in this particular case. You can imagine the consequences of that, of malaria parasites accumulating in the placenta. The impact on the developing baby is very serious and can lead to death and low birth weight. There are also inflammatory responses that set up a raft of sort of effects that lead to disturbances in the way the body functions. And there's direct destruction of red blood cells, because malaria parasites infect red blood cells, develop inside them, and then they rupture, as you saw in the video. But also, for reasons that we don't clearly understand, that malaria causes the body to destroy its own red blood cells. So you can get very, very severe anemia. All of these things are operating together and lead to a severe illness which has this multi-system involvement. And by that I mean it involves multiple different organs and different systems, your circulatory system, neurological system, kidney function, lung function, and so on. So they're the sort of main features, as I highlighted, that we see in individuals. So to treat malaria, mild malaria is actually pretty easy to treat. As I said, it's a flu-like illness. If people can actually get treated, get access to medical care quickly enough, they can take a short course of effective anti-malarial tablets, oral medication, and there are a number of treatments that have been used over the years. Chloroquine, 3 days treatment, very effective until the last probably 20 years, when resistance has become such a problem that that drug can no longer be used. Also very cheap. A drug called SP, sulfadoxine pyrimethamine, quite a remarkable drug in many ways. 1 dose, 3 tablets in an adult, and essentially 95% cure rates, until again the last 10 years particularly, 20 years, when we start to see increasing resistance, and in many countries they're now having to discontinue the use of that drug because of drug resistance. Then more recently, drugs such as artesunate, which you can take for 7 days, 1 tablet a day for 7 days, very effective, 90% effectiveness, efficacy, and that's derived from a plant that was developed through Chinese medicine, and is an extremely effective anti-malarial. So that's mild malaria, pretty easy. The problem is severe malaria. It's very difficult to treat, particularly when someone presents to a health service and they're critically ill. You obviously need a lot of facilities to treat people in that situation, and those sort of facilities are typically lacking in countries where malaria occurs. So first off, you've got to give people anti-malarials, and this needs to be intravenously. And particularly artemisinin drugs like artesunate have been shown through clinical studies across the globe to give people a much better outcome. So usually IV, intravenous for 7 to 10 days, sometimes longer. The problem is, what else do you do? Because still a proportion of people will die, probably 30% in Africa, even if you get anti-malarials into people as soon as you possibly can. So, we use intravenous fluids, blood transfusion if required to correct anemia, supportive treatment essentially, such as you would find in intensive care or high dependency units here, obviously at a more basic level in many places. But things like anticonvulsants to control fits and convulsions have been tried, anticoagulants to try and correct blood clotting disorders, even anti-inflammatory drugs, experimental and established, things like cortisone. On the whole, most of these have had a fairly disappointing effect. And there's little evidence that these have, really play a major benefit to people with severe malaria. So that really highlights the point actually that over decades there's really been little improvement in the mortality rates of severe malaria. So, in Africa, about 30% of people with severe malaria will die, and we've really not made much progress over the last probably 40 years in trying to get that number down, because we need specific interventions to target the processes that are occurring. I should also highlight, though, that the majority of people who die of severe malaria around the world don't do it in healthcare facilities. They do it at home, or they do it in a setting where really only very basic healthcare facilities are available, and the sort of interventions needed are just not available. And that really emphasises the need for prevention, things like vaccines and control measures to really reduce the burden of disease and prevent it, rather than trying to deal with the problem of severe malaria. Just to talk a little bit about immunity, how can we sort of, in the context of trying to prevent malaria, what do we know about immunity, and how could we try to use that information to develop vaccines, for example? Well, there are two types of immunity and resistance to malaria. The first type is a genetic one. The example of sickle cell trait, Alan's covered. There's also alpha thalassemia, another haemoglobin disorder, and actually a number of major and minor blood groups that influence people's susceptibility to malaria. So there's not much you can do about that. And there's not much we can do about that. It's just what's evolved over a millennia, really. But what's particularly important to us is to understand acquired immunity. So after repeated exposure, after people have many episodes, potentially 20, 30 episodes of malaria, if they survive that, they do actually develop significant immunity. And that immunity controls parasites, prevents them from replicating to large numbers in the bloodstream, and prevents the development of severe disease, and in many cases, any symptoms at all. So we think that understanding how immunity develops and what the immune system targets is a key to developing an effective vaccine. So that sort of leads into the work that we do, and I just wanted to briefly cover some of the research that we do that's particularly tailored towards trying to develop interventions to combat malaria. So we're particularly interested in malaria in pregnancy and malaria in early childhood, which are the two times when people are most susceptible to malaria, at greatest risk. A lot of our work is focused on the development of vaccines and new treatments, and we also have some involvement in things like clinical trials. These are studies designed to try and test new treatments or new interventions. A really important part of what we do is building strong partnerships with researchers and institutes in malaria endemic countries. So that's a really important part. As Alan highlighted, this is a disease that affects mainly resource-poor countries, and it's not a disease that's prevalent in Australia, obviously. So just two examples here of collaborators are Kenya Medical Research Institute, which have these fantastic new facilities funded by the Wellcome Trust in coastal Kenya, where malaria is a huge problem. And this is just an example of the really excellent new labs they have that allow them to do state-of-the-art research in a resource-poor setting. This is a student who was working with me, working together with a Kenyan-trained scientist. And this is some pictures from the Papua New Guinea Institute of Medical Research, which is also an extremely important partner in our battle against malaria. Although less fancy facilities, they've been able to do some remarkable work over the past several decades. This is just an example of one of the labs and an example of a research clinic looking at trialling new treatments. And I'll just sort of emphasise that malaria affects resource-poor communities and really the poorest of the poor, because, as I think you probably got the impression from Alan, and you probably know already, that the most affected countries are developing countries, countries in sub-Saharan Africa, South Asia, Southeast Asia. And it's rural communities that are affected. And these are the communities that have the least access to medical care, the lowest incomes, and the lowest resources. There's just some typical village scenes from Africa. A typical village in Africa, there's no running water, there's no electricity, there's no postal address, and so on. And these are just two examples of healthcare facilities in sub-Saharan Africa. In this case, I've used Malawi as the example. So this is a traditional birthing house where many women will give birth. These are traditional birth attendants. They have very, very little formal education of any type. no running water, no medications available, no electricity. This is an antenatal clinic of a regional hospital. It's only an hour's drive from the major, not capital, but the major city in the country. Again, this has, when I visited, this has no electricity, no running water, and it's remarkable, that you just can't run provide quality care for pregnant women under those sort of conditions, and that's really typical throughout, most regions where malaria is occurring. So that really, again, just comes back to that point, that prevention and finding ways to prevent and finding interventions that can be rolled out across populations is absolutely crucial. There are other barriers, such as physical barriers. This is a Karen refugee camp in Thailand. You can see the terrain makes it very difficult to access. There's political and social issues as well, which make it difficult. But, there's a lot of goodwill and a lot of political commitment to trying to find solutions to malaria. So just as an example, in PNG, was this a real focus for us, partly because of the links that we've built up over, probably 20 years or more, actually. Possibly 30 years even, between the Hall Institute and the Papua New Guinea Institute of Medical Research. We have programs looking at preventing malaria in infants and young children and pregnant women, programs to understand how malaria disease develops that would lead to better treatments, and programs to understand how immunity develops to help us work out how to develop a vaccine for malaria. There's just some examples of a clinic, research clinic with some research staff from the institute and a typical hospital that some of the projects worked out of. I think working in PNG for people on these projects has its rewards and its challenges. I mean, this is a typical sort of scene from a village meeting, and these are a really important part of involving the community in the research. But obviously access can be a real challenge. These are some examples of clinics. And so one of the ways we understand immunity is to follow children over a period of time, and in this particular case, followed 200 children over 6 months. Nearly all of them became infected, but only a proportion actually developed illness. So about half got sick, the other half got infected but didn't get sick. So clearly those kids who were controlling the infection are really interesting to look at, because they'll provide us insights into how the immune system is controlling the disease. And we think that antibodies act by blocking infection of red blood cells. So just lastly, to touch on some work on developing a vaccine. This is just briefly the life cycle. So Alan's gone through this already, but one approach is to target this sporozoite form that's injected by the mosquito to either block the entry of sporozoites into the liver or inhibit their development and therefore stop them getting into the bloodstream. The other way is when the parasites come out of the liver, find a way to block their ability to infect red blood cells and prevent replication. Something that Alan and others at the Hall Institute are working on is, is it possible to develop a live attenuated vaccine? That's a vaccine in which the parasite is genetically modified so that it's sort of weakened, and this weakened parasite can infect the liver but can't develop properly. The idea is that it would generate an immune response without leading to disease, and this would be an effective vaccine. So this approach is going to be tested in volunteers in a controlled environment first to see how this would be able to be developed. The other approach I'll just show you very quickly is a strategy to try and block that infection of red blood cells by the merozoite form of the parasites. And this is just a video of looking at parasites under the microscope. This is a merozoite form getting into a red blood cell and crumpling up that red blood cell as it does so, and then it will develop over a period of 2 days. And you can see this is counts in seconds, so it's happening over 2 or 3 minutes. Although this process happens very quickly, it can be blocked, and you can see here merozoites buzzing around, coming into contact with red blood cells, but not getting in. So in this example, we've used a compound which blocks that process, but it's also possible to achieve that with antibodies generated by vaccination. So I won't cover this, but it's really just to highlight that, it's not really to go into any detail, but to highlight the point that if we go from a population level to looking at understanding immunity, look under the microscope to the sort of mechanisms that are occurring, understand the molecular interactions and understand the molecules, the proteins the parasites use, we need all of these strategies to really tailor an approach that's going to effectively target malaria parasites and neutralise them, stop them replicating in the bloodstream. So just to sum up by saying, what's on the horizon, what are we looking at in the future? Well, clinical trials of new vaccines, the sporozoite live attenuated vaccine, first trial is starting later this year. We hope to start some initial vaccine trials with some of the merozoite antigens in 2 years' time. And development of new drugs for malaria is really a long-term investment. There's a lot of identification of key targets, potential compounds that are then further developed, and there are several promising compounds that have been identified that that the researchers here are working on. Okay, I'll leave it there. Thanks.
Well, thank you, James, for covering the malaria as a disease, some of the strategies being used to develop new tools to control infection and disease, and also, very importantly, highlighting those collaborators which WEHI sort of works with in these disease-endemic countries, and, of course, the realities of the situation where malaria is occurring. So thank you very much, James. Our third and last speaker is a former director of this very institute, Sir Gustav Nossal, universally known as Gus Nossal. He's Australia's best-known medical research scientist and advocate. For 50 years, his research interests have centred around the cellular events of antibody production. But his scientific achievements are matched by the energy he has devoted over his entire career to public health, science in society, political, philanthropic and community support for science, global access to existing vaccines and development of new vaccines, the sort that James was just talking about here, and also the personal and scientific development of colleagues and young scientists. He is sort of a mentor to many of us. I know for a fact that he's had a very deep interest in malaria for exactly 33 years, Gus, as I calculated, and we look forward to his presentation and his insights as our third and final speaker, Gus Nossal.
Thank you, Graham, for those typically over-generous remarks. And might I say, it is a real delight to see this room full to overflowing. What a vindication of the idea of these public lectures, which clearly show the community is very interested in what the Walter and Eliza Hall Institute is doing. Now, my job here is to embed the research in malaria in a kind of a global context, and first of all to ask the question, what is the world as a world doing about third world diseases? And you'll be interested to hear that in 2008, the world spent $3 billion US on research and development for third world diseases. Now, you'll immediately say, that's a lot of money, $3 billion. Wow, aren't we good? Well, I have to tell you that in that same year, one country, the United States alone, spent $90 billion on medical research and development, 30 billion government, and 60 billion industry spending. And by the way, that ratio is a fairly general one. When you think about R&D, don't just think about governments. Private companies are very, very heavy investors in R&D. If you now think about the world, the total world figures for R&D spend is about $200 billion on medical research, medical and health research. Now, in WHO, where Graham and Alan, many of us spend a lot of time, they have this little happy catchphrase, the 90-10 gap. 90% of the world's research funds are spent on diseases which 10% of the world get. But actually taking these figures out from the web a couple of nights ago, I realised that it's underselling it, because that 3 billion based on the 200 billion is in fact 1.5%. It's not 10%, it's 1.5%. And so we're really undercooking research in the third world. For those of you that are interested in figures, you'll be interested what the global spend on total R&D on all subjects, and that is in fact 1.6 trillion US dollars, 8 times more than what's spent on medical research, again, largely by corporations, 2/3 of it by corporations, and that represents about 2% of GDP. And you'll find now in discussion paper after discussion paper this figure, what percentage does your nation spend on R&D, being used as a measure of the likely success of your nation in the future. Now back to medical research, and back to medical research in the third world. 60% of the funding, that $3 billion, comes from just two sources: the US government, largely via its National Institutes of Health, and the Bill and Melinda Gates Foundation. European and UK governments come a distant third, 12%. And interestingly, innovative developing countries, you've heard of the BRICS, Brazil, India and South Africa are becoming sufficient players, spending about 4% of that 3 billion. I should also mention, seeing as we're talking about tropical diseases here, the tremendous contribution down the decades of the Wellcome Trust, which is the second largest medical research foundation after the Bill and Melinda Gates Foundation. Now, one interesting thing has happened. The George Institute in Sydney has recently done quite a lot of research on just what do we spend this money on. And it turns out that the lion's share goes to just 3 diseases: HIV/AIDS, 39%, malaria, 18%, and tuberculosis, 15%. And of course, from the point of view of today's seminar, that's great. We've got lots of interesting new research in malaria to tell you about. But actually, if you want to be a global citizen, you will find that diarrheal diseases and acute respiratory diseases are seriously undercooked, and they, of course, cause a lot of mortality as well. Now, the challenge of malaria. You've heard that there's at least 300 million attacks a year and at least 1 million deaths, 90% in Africa, mainly in kids under 5 and in pregnant women. After the age of 5, this tenuous partial non-sterilizing immunity develops. I want to say a word about Vivax, because of course all of that has been falciparum. Vivax is what gives malaria its bad name in many ways, because if you haven't taken your drugs, your prophylactic drugs, and you do get an attack of Vivax, which as a traveller you're quite likely to do, it can come back, it goes underground and come back years later. So it's given this bad name, which falciparum doesn't do that. It either kills you or you recover from it. Now, I'm pretty keen that those of us who think about malaria vaccines think more about Vivax, because frankly, I think in the long race that there will be till we get to the end goal, we'll be very reliant on rich travellers. And the rich travellers will want to be protected against Vivax as well as falciparum. Now, you heard that the malaria parasites are developing resistance to the first-line cheap drugs, and you probably know that the Anopheles mosquito vectors are developing resistance to the common insecticides. And you have heard, but it's worth reinforcing, that malaria constitutes an economic burden with the high cost of treatment and the decreased productivity, as well as a humanitarian health burden. Now, I want to say that despite all of these challenges, there are reasons for hope. And a medium tech, I'm almost tempted to call it a low tech intervention, holds pride of place here. Insecticide impregnated bed nets at a cost of approximately $5 can actually reduce childhood mortality by at least 50%. And that's aided and abetted by indoor spraying of those primitive dwellings that you saw. And drugs, despite the constraints, as James has told, you can both prevent and cure. And a bold private sector, public sector partnership, which is called the Medicines for Malaria project, is developing new drugs. Altogether, there are now 39 R&D partners, not just the WEHI, great though it is. There are actually 39 R&D partners in this consortium. This was started by Gro Harlem Brundtland, the former Prime Minister of Norway and best known for being the chairman of the Sustainable Development, that great report from Rio, which got sustainable development onto the agenda. She became Director General of WHO, and I think in her 5 years, the biggest thing that she did was to persuade those bureaucrats in the organisation to look more kindly on industry. Now, industry is the whipping boy. Industry is the one we all love to hate because they're making money out of human misery and all of that kind of stuff. Well, she thought that industry had to be a player in this whole game of ridding the world of these diseases, and she was very much aided and abetted by Sir Richard Sykes, who was then the chairman of what is now GlaxoSmithKline, then Glaxo Wellcome. And the best single thing that they did was the Medicines for Malaria partnership. Now, I want to say a bit about the vectors. This is a little bit space age, but you'll be surprised, and largely funded by the Gates Foundation, there's a lot of advance in vector biology and control. For example, the thought of genetically developing sterile male mosquitoes that, however, despite their sterility, are super males and therefore can compete with normal males in terms of fertilizing the female mosquitoes. And that has great promise. The other approach that's making quite rapid strides is developing female mosquitoes through genetic engineering that are incapable of supporting parasite maturation. That parasite maturation was so beautifully shown in the video and so beautifully talked about by Alan, takes about 5 days. And so if you can have a mosquito that'll die 3 days into the process, or that will be in some other way altered so that it can't support that process, you have a powerful weapon. Now, I don't want to hide from you that there will be huge difficulties in making this into a practical matter, the difficulties of large-scale release and the ecological considerations of what unexpected things might occur if you release strange mosquito strains into the world. But you should know it's a very exciting area of research. Now, I too want to say a little bit about malaria chemotherapy and to pay some tribute to 2,000 years worth of research in China, because artemisinin is a Chinese discovery. They discovered the sweet wormwood tree would make this tea, which was a powerful anti-malarial. And they alone isolated the active principle and gave us artemisinin. And also, while artemisinin is the best single drug that we have, artemisinin-based combination therapy in combination with some of those other drugs that I've listed have a great virtue. Now, how many sweet wormwood trees are there in the world? How many can we grow? It's a pretty similar thing to what we have with the great Australian drug Taxol, which is grown from gum trees in Western Australia. The answer is there'll be a limit, and the answer is a lot of work to make a synthetic artemisinin. But would you believe that despite all the effort, we're not quite there yet? There's been very good progress made, but this is not yet a synthetic drug. Now, intermittent preventative treatment, or IPT, with ACT, with artemisinin-based combination therapy, is a great public health measure. And I'm a bit old-fashioned here, James, going with the old SP, sulfadoxine pyrimethamine. But if you give this to asymptomatic patients, asymptomatic infants, in high risk areas, you can materially and seriously lower the mortality from malaria. And IPT is also effective in pregnancy when usually 2 courses are given. Now, there are people who espouse great hopes, and I suppose the greatest hoper in the globe is Melinda Gates. And Melinda Gates was very, very active in urging the United Nations and finally having enacted a global malaria action plan. On the 25th of September in 2008, a special session of the United Nations launched a summit, a $3 billion plan to, quote, end all malaria deaths by 2015. Well, the failure to develop a malaria vaccine was a very serious flaw in Gus Nossal's otherwise stellar career. But I'll confess to a smaller failure. I tried very, very hard to stop the Gates Foundation from using the E word. Because we've been there, done that, and been once bitten. I think eradication of malaria by 2015 is a goal, the likelihood of which is very small indeed. And I would have much preferred them to use some phrase like the elimination of malaria as a major public health threat or whatever. But I lost that one, and Melinda keeps talking about the eradication of malaria. In any case, for better or for worse, they've pledged a lot of new money towards new malaria vaccine research, including that which is helping to fund Alan's team's outstanding work. Another thing that's happened out of the initiative is the pledge for 100 million of these insecticide-treated bed nets. Alan and I had a pretty interesting experience last year. We went to Arusha, Tanzania, and yes, Gates likes to fly the flag, and they have the meetings often in third world countries. The major meeting, the previous one to that, was in Cape Town. And there in Arusha, Tanzania, I can take you to a factory that occupies 3 times the area of the MCG and that employs 7,000 people. What are those people doing? From beginning to end, they're making insecticide-impregnated bed nets for the world. And that's where the 100 million are going to come from. So that's pretty amazing. Now, everybody likes to knock multinationals. I do it from time to time myself, although I'm often clad or described as an apologist for the drug industry. The truth is sort of somewhere halfway in between. But really, GlaxoSmithKline deserves very great credit for their work towards a sporozoite-based malaria vaccine, which they call RTSS. Together with the Walter Reed Army Research Institute of the United States Army, in other words, the US Department of Defense, they have spent, wait for it, 25 years developing RTSS. And by my count, which may not be entirely accurate, that involved no fewer than 20 clinical trials, the first 19 of which failed. Finally, they came up with this RTSS thing, which has passed its phase 2 clinical trials with not a very exciting efficacy, 50 to 60% efficacy in infants, and probably too short a duration of immunization. But let me say it's been a ray of hope in a very difficult area. And they are being helped by the Gates Foundation to go through the phase 3 trials now, 16,000 infants in 10 different sites in 7 countries. And as Alan has said, we will, or maybe it was James, at the end of 2011, we'll have some idea of whether RTSS really works. Now, I think we all know that RTSS will not be the final malaria vaccine that the world will use for the rest of time. And I'll take you back for the third time in this symposium to this life cycle slide because it's absolutely essential. And we'll just go through it slowly now to have a look at what might be being done and what might be the eventual final malaria vaccine. RTSS acts on this first stage, the sporozoites, that the mosquito injects under the skin, into the skin in the human host. The live attenuated approach, which you have heard, comes in 2 iterations, and the RTSS, I should say, is a molecular vaccine. It comes from the molecules that are coating the outside of the sporozoite. The live attenuated uses the whole sporozoite. And why is one daring so much to talk about that? Well, interestingly enough, it's an approach that we know works. We know it works. We've known for 30 years that if you irradiate sporozoites and inject them into a person, or irradiate a mosquito and allow the irradiated mosquito to feed on a person, you will cause immunity to an attack of malaria that you will give that volunteer. So we know that it works. However, the funny thing is, can you actually imagine a situation where there are enough people milking salivary glands from living mosquitoes to immunize the 6.7 billion citizens of spaceship Earth? Well, there's no one in the world crazy enough to try that other than Steve Hoffman. And you would have to know Steve Hoffman to understand it. He burnt his fingers very badly on a molecular vaccine while he was with the US Army. But he has persuaded the Gates Foundation to give him $300 million for a firm called Malaria, which is indeed proving whether or not irradiated sporozoites from irradiated mosquitoes and milk from the salivary glands can constitute an effective vaccine, and he is in clinical trial. However, Alan Cowman is much smarter than that, and he says, I don't want to fry my vaccine, and God knows what other damage I will do to the sporozoite by the harsh treatment of X-irradiation. So with Stefan Kappe in Germany and his team, Alan has been funded to try to develop a genetically modified live attenuated vaccine. And we also, of course, have to think of other ways of growing these sporozoites other than having them in the malarial salivary gland. And that's a difficult exercise in tissue culture. Now, that's the first line of defense. Second line of defense, you've already been told, are these liver cells. But what James didn't have time to tell you is that Rolf Zinkernagel and Peter Doherty, our own Australian Peter Doherty, won the Nobel Prize for figuring out that there's a second way that you can have immunity other than just antibodies. They figured out that, in fact, on the surface, in this example, of these liver cells infected with sporozoites and now harboring the growth of these merozoites, there are little portions of the parasite that are popped onto the cell surface in association with very specialized presenting molecules. And that makes a juicy target for a different kind of lymphocyte that Graham Mitchell and Jacques Miller discovered, called a T lymphocyte. And it does not act like an antibody, which is like a sort of a rifleman shooting a bullet right across the room. It actually throttles the cell to death. It delivers a dagger right into the liver cell. And if we could get a T-cell-based vaccine against antigens of the liver, of the parasite living in the liver cell, we'd have another approach to aborting this cycle. The leading worker in this field is Chetan Chitnis in the Institute for Cellular and Molecular, what's it called? ICGB, International Centre for Genetic Engineering and Biotechnology in New Delhi, India, and of course he's a great colleague and collaborator. So a liver cell, a component to the parasite. The Hall Institute's work down the decades has mainly been, as you have heard, on the blood stage of the parasite. And we had two reasons, Graham and I, for making that decision. The first is that we had so much respect for Ruth Nussenzweig, who discovered the sporozoite antigen. We said the sporozoite antigen is going to be in good shape and won't need us to work on it. And secondly, we said it's the blood stage that makes you sick, and we wouldn't mind a vaccine that, if it sort of half worked, at least made you a little less sick. That work is ongoing. There are a number of very, very interesting molecules, including one that was discovered by Robin Anders and Ross Koppel and Graham Brown called AMA1. But I should also say we've been fortunate to be joined in this effort by La Trobe University and Monash University, and the Australian effort towards a blood stage malaria vaccine is very much alive and well. Finally, as my last point, I will mention the unselfish vaccine. Right down at the end of the process, you will see these gametocytes, the ancestors of the two gametes, the male and the female gamete. And yes, the gametocytes have special antigens. If we can inject humans with antigens from these sexual forms, we can create a situation where these gametocytes enter the mosquito coated with antibody and can't complete their life cycle. This is the unselfish vaccine. It will do absolutely no good to the individual injected person, because the gametocytes don't make you sick. But on a community level, if this can be made to work, you will have very much less transmission. And, futurology is a difficult art, but when you're just a couple of weeks from your 97th birthday, it's not as scary as if you're James Beeston's age. So I think I can confidently say when the world has its definitive malaria vaccine by about 2050, and it will take that long, it will have components of all of these four stages: sporozoite, liver stage, blood stage, and gametocytes. Well, ladies and gentlemen, the talks ranged a little bit over medical politics and a little bit over science, and it's difficult to resist the temptation of calling on Mac Burnett's fellow Nobel laureate, Sir Peter Medawar, the father of transplantation biology, who coined the phrase, politics is the art of the possible. But in fact, the full quote is, if politics is the art of the possible, research is surely the art of the soluble. Both are immensely practical-minded affairs. So what he's trying to tell us here is that yes, your applied research is possible and creates new tools, but the basic research which underpins it, the research, the art of the solve, what problem can I solve, is immensely important. But seeing it's vaccines that we're talking about, I can't refrain from leaving the very last word to Louis Pasteur, because this word, of course, describes the life of all the medical researchers in this room. None of us want to be known as just good researchers in Australia, because, as Louis Pasteur said, science knows no country, knowledge belongs to humanity, and it is the torch which illuminates the world. Thank you.
Thank you very much, Gus, for that inspiration. Now, you've been very, very tolerant. I hope you've enjoyed these presentations. I think there will be some who must leave at 7:30. It's perfectly okay to up and leave, but I think we'll eke out 10 minutes of our refreshment time for your chance to delve deeper into some of the issues that we've got three experts here is a chance to get some additional discussion going. So, colleagues, if you wouldn't mind coming to the... Okay, as I say, we will break for refreshments and continue the conversation in 10 minutes from now, but the floor is open. Our panel is available. Yes?
Have they ever eradicated malaria from any place in the world?
Yes, we have. Some good examples are Europe, Spain, Italy, even England. That had malaria in the past. Parts of the former Soviet Union, the countries encompassing that, and some islands, Taiwan, malaria was eradicated. So yeah, it has been possible, but mainly where malaria has been sort of marginal. In Africa, sub-Saharan Africa, and PNG, a whole range of reasons make eradication extremely challenging, even if we have this resources available.
We thought we had the tool with DDT was going to be the basis of the malaria eradication in the 50s. There was one lesson to be learned from that. We thought we had the tool, and we sort of downplayed research in malaria because we were applying what we know now. Total disaster. Unlike the smallpox, though, yes, they had the vaccine, but they invested maximally in research because they knew there'd be questions coming back from the field in terms of implementation of the strategy, which needed a sort of researchers to get groups of. We don't have smallpox, we do have malaria. Yes?
I'm interested in hearing from the panel the views on the impact of climate change on the spread of malaria and whether that's impacting on the research efforts, particularly in the private sector. I totally agree with Sir Gus's comment about Vivex. I got it 20 years ago and I live with it and am very aware of it. But, is that having an impact on the research effort?
If we look at Australia, with the effect of climate change, obviously it'll become warmer and perhaps wetter in the north and more conducive to mosquitoes that carry malaria. I think for a developed country like Australia, it will be possible to control that in the future. So I don't see any issues with the spread of malaria to a country like Australia as the world warms up. It may have a negative effect with respect to countries like continents like Africa in terms of the level of malaria, which may actually decrease with alterations in climate. Malaria parasites, as I think we know, can evolve very rapidly and take a mosquito. So whatever changes thrown at it, I think they will evolve to occupy new niches in whatever climate.
It was always said that Australia would have a lot of malaria if it was built upside down. Sydney and Melbourne and the rest were up there. Yes.
We're not that far from New Guinea.
We're not that far from New Guinea, and the vectors will sort of change their range and distribution, sure. Yes?
I just wondered whether human immunoglobulins were ever used as a form of treatment.
Well, there's a lovely study that's about 50 years old that shows that antibodies can work in an actual attack of malaria. But are not of these just proved in the development, James?
Yeah, I think there are some examples, some studies done as proof of principle. But as a form of treatment, it's had a lot of side effects. It's difficult to do. And much simpler ways of treating people. That would be anti-malarials that are available. The other problem is now, of course, we've got much greater awareness of blood-borne viruses, HIV, hepatitis C, hepatitis B, and others. And it just would never be achievable to do that in malaria.
Yes.
What makes some mosquito species so good as vectors and not others?
That's a very good question, and it's not one that has an answer at the moment. It's an area that a lot of people are researching, trying to discover the genes that are really important in terms of transmission for mosquitoes. So at the moment, no one can really answer that. Some of the genes or proteins that are important for transmission of malaria have been discovered. There's a couple that are known at the moment. And some mosquitoes don't have the same form of those particular proteins. They have mutations that essentially blocks the malaria parasite from being transmissible in those species. If you put those genes into those other species, the ones that work in particular species of mosquito, and ask, do those mosquitoes now, are they now able to be transmissible to mosquitoes? The answer is usually no. There's probably multiple mutations in multiple genes and proteins that are required. So at the moment, a lot more research is needed in the area.
1, 2, 3. Yes, go ahead.
I was just wondering why the parasite doesn't actually affect the mosquito at all. It seems to just grow inside the mosquito and then just get reproduce inside the mosquito and then get passed on. It doesn't actually make the mosquito sick at all.
Another area of active research, and the answer is, in fact, not many experiments have been done to really answer that, but where they have been, it does affect the mosquito. The mosquito is not as healthy, the infected mosquito is not as healthy as an uninfected mosquito. So it does have an effect on the mosquito.
But it doesn't kill the mosquito.
It does.
They just get a bit sick.
I think more research needs to be done on that. Certainly, it probably affects them enough that some die, but not all.
One of the issues is that the studies are done in a controlled environment, and probably the death rate of mosquitoes in a natural environment is much higher. So there's some suggestion that they're less they are infected. But how that translates into the death of mosquitoes in a natural setting is hard to determine.
It'd be nice if they were really slowed down and they were taken by a bat or a swallow. Yes.
Can one mosquito harbor the different species of the Plasmodium? And is there any interaction if a person or if an individual has several different Plasmodium?
There's not many places. In Africa, for example, the vast majority of malaria is caused by one species, Plasmodium falciparum. But in some places, particularly say Papua New Guinea, you get sort of equal amounts of Plasmodium falciparum and Plasmodium vivax. And in some other places in Vanuatu, for example, and Southeast Asia, you sometimes see that. You see mixed infections. They're the two most common that go together. However, it's not total. There's not sort of total consensus from all the studies what the end result, what the end effect is of having the two together. Is it worse or better than having one alone? And there's studies that show both ways. Some studies suggesting that it's actually better, and some studies suggesting that you get some sort of cross protection, but others that haven't found that. So I think it's hard to dissect.
Alan, will your attenuated sporozoite vaccine have any cross protection, or do we know before we start that don't even bring it?
No, I think we should dream of it. I think it's unlikely, but it's possible. There may be some cross protection because there's some conservation of energies.
Yeah.
So,
giving it so many, there has to be some.
So I think it's possible and worth finding out.
Yes.
I gathered from Mr. Gus's talk that it will be about 40 years before the vaccine is finally available worldwide. While we are waiting, it occurred to me that why don't has any research been done about insecticides? Say the DDT program failed, chemistry has advanced. Can't they find some other insecticide that we can use as an interim measure?
I didn't want to leave the impression that there won't be a malaria vaccine at all. In fact, GlaxoSmithKline are very keen to introduce RTSS, which is partially effective vaccine, as soon as the clinical trials are over and as soon as the regulatory agencies give it the information and it becomes a licensed vaccine. And that could easily be 2013, 2014. But what I'm saying is it won't be the final answer. The final answer will take much longer. My ideal vaccine is 4 stages will take much longer. Now, in the meantime, yes, there is a lot of work on new insecticides, using that word in the broadest sense. There's a big GATES program which is looking both at chemoattractants and chemorepellents, and try to have new drugs in both areas. Chemoattractants to pull a mosquito into a trap of some sort, chemorepellents for your personal protection in the impregnated bed nets. Some of that work is quite fascinating. When some of us go to the GATES meetings, it's rather fun to note that the positive control which scientists use for the best chemoattractant for mosquitoes is smelly socks.
Yes, let's have 2 or 3 more. But we're getting a little wind up. One, maybe 2 questions. Yes.
How can you insert the PFENP1 protein between the 5 species? And are there any possibilities of making a synthetic blocker or inhibitor?
Yeah, so PFNP, Plasmodium falciparum is essentially the only one that has PFNP1. So it's been evolved fairly late in the evolutionary history of malaria parasites. In terms of how conserved it is across the Plasmodium falciparum. There are regions that are conserved quite well, but it is incredibly diverse. The parasite has mechanisms to change it rapidly, because antibodies are produced that can essentially destroy the parasite. So it has mechanisms to alter its surface coat, switch to new ones that the human immune system hasn't seen before. It's a system called antigenic variation, very classical for lots of different organisms, infectious organisms. So in terms of what's the likelihood of making a vaccine using PFNP1, unlikely, except for one case, one example, and that's which affects pregnant women. And perhaps, James, you can add a bit more to that.
Yeah, sure. There's one particular variant of that group of proteins that appears to be important in pregnancy and enables the parasites to accumulate in the placenta, which I showed in my talk. And that particular variant seems to be either more conserved and have less overall diversity, or have conserved areas that people have been able to target vaccine development. So that's one line that people are pursuing. I think realistically, for that group of antigens, that approach might be successful for pregnant women, but I don't think it will be. It's probably unlikely to be successful as a sort of broad vaccine for everyone.
Thank you for that question, Lydia. Any final question? Yes.
Just one. I didn't understand the infection numbers. On the one hand, you've got 3.3 billion people exposed to malaria, I think you said. And yet, in the study you did at PNG, you had 97% of kids were infected in 6 months. So if that's the case, how come you only have 250 million cases of malaria a year? Is it feasible that a whole bunch of people out there don't get infected, they're contaminated but don't get infected, they have some sort of, not resistance, not immunity as we know it, but they're just not affected? Is that possible?
Yeah, I think the key thing is there in those calculations, when you look at the global number, is that a lot of those people have been exposed many times, so they've developed immunity. So if you've got a population of 100 million, well, the adults will no longer be susceptible because they've developed immunity if they've survived. So that's sort of why the numbers don't necessarily all translate into just sort of simple calculations, because the underlying factor there is people are acquiring immunity as well, so their risk drops. So you have to sort of look at the population of kids really that are the highest risk.
Okay, well, I think on your behalf I'd like to say thank you to our panel members. Can we say thank you for your participation? Now we adjourn for a refreshment.